So we're going to start out this video by talking about something that I know you'll feel totally comfortable with listening to, and that is types of birth control. And before I even get started, I need to say that I don't advocate for or against any of these types of birth control. I'm just going to talk about what they are and what the advantages and disadvantages may be. So we start out with abstinence, and this is basically not having sex at all. And of course, that's 100% effective, and it's free. It says the disadvantages are that it's difficult to do, and so I'm just going to kind of leave it at that. A vasectomy is another type of birth control, and this is where the vas deferens, each vas deferens, is cut. And so the advantages to this type of birth control is that it's permanent, and also it doesn't interrupt spontaneity. You don't have to stop what you're doing and administer any kind of birth control. The disadvantages are that it requires minor surgery, and yes, it actually is minor. It's outpatient surgery. It heals up pretty quickly, but it's difficult to reverse. So once that's done, it may not be reversible. Withdrawal is another birth control method, and this is the removal of the penis from the vagina before ejaculation. There's no cost to this, but there are a lot of disadvantages to this, and it's not recommended for people who are sexually inexperienced. It requires a lot of self-control. Sperm may leak from the penis before withdrawal, so some can come out before ejaculation. And if the vulva is ejaculated on, it's not even in, but on, it may cause pregnancy. And this figure says shows pregnancies per 100 women per year. It says 18. But there's an old joke that I've heard that says, what do you call people who practice withdrawal? Parents. So it's not something that I would recommend as a reliable means of birth control. Next up, we've got latex or polyurethane condoms. And the mechanism is basically it's a sheath that's worn over the penis. There are some female condoms that are inserted into the vagina, but the whole point is this is a physical barrier that keeps sperm out of the vagina. And so an advantage is that it protects against sexually transmitted diseases. The disadvantage is that it disrupts spontaneity and reduces sensation. But, you know, let's be honest, would you rather have spontaneity or a kid or disease? Next up, we've got a cervical cap. I just said that I wouldn't advocate for any of these. I'm sorry. I guess it sounds like I just did. A cervical cap can be used with spermicide, and basically this is a physical barrier that just blocks the cervix. And with spermicide, it'll actually kill sperm. So the advantage is that it's inexpensive, and it can also be kept in for 24 hours. So if you're anticipating something happening, then it can be used in advance. But it may slip out of place. It has to be kind of custom fitted, and it's a lot less effective for women who have already gone through childbirth. And you can see that the pregnancy rate is even higher than that of withdrawal. An IUD prevents an embryo from implanting, and so here's our advantage of not interrupting spontaneity again, but there are some disadvantages. It can cause severe menstrual cramps, and there is a risk of infection. It can also cause pelvic inflammatory disease. So this is basically something that irritates the endometrium to prevent implantation. Tubal ligation is where the uterine tubes are cut, and the reason why for this is so that oocytes don't reach the uterus. And it's basically the same thing as a vasectomy. It's permanent. It doesn't interrupt spontaneity. Now, the surgery is not as minor as a vasectomy is. So there's additional risk of infection, and it's also very difficult to reverse. But like a vasectomy, it's got a very low rate of pregnancy per 100 women per year. So another is the fertility awareness method. This is sometimes called the rhythm method. And so this happens when there's no intercourse during fertile times. Body temperature is a clue as to ovulation, and there are other clues as well. And so the disadvantage is that this requires careful record keeping. But the other thing is the menstrual cycle is not always completely consistent from month to month. And so there is a pretty great risk of pregnancy here, 1 in 5. And so I've heard the same joke with withdrawal also applied to the rhythm method. What do you call people who use the rhythm method? Parents. Okay, hormonal contraception involves basically the introduction of estrogen and progesterone. And what this does is to hormonally mimic pregnancy and prevent ovulation and implantation. And so this says it's easy to use, but you do have to keep careful track of this. And what are the disadvantages? Menstrual changes, there can be weight gain, there can be headaches. Basically, if you disrupt somebody's menstrual cycle like this hormonally, it can cause a lot of the same kind of fluid retention and mood swings. And for some women this works very well, very smoothly, and for some women it makes them absolutely miserable. And what are the pregnancies per 100 women per year? Between 2 and 9. And so what the 2 asterisk statement at the bottom says that for some methods the number of pregnancies may be much lower if the method is used perfectly every time. And so I think that that some of the pregnancies occur if somebody forgets to take just 1 pill per month with the pill. So if you forget 1 pill during the month, then you have to start all over again the next month. And so that does require careful record keeping and tracking. So the methods mentioned in the last slide of birth control that involve a physical barrier can also help prevent STDs, sexually transmitted diseases. And so how can you prevent the spread of STDs? You can abstain from sex. You can have only one sexual partner, this assuming that the sexual partner doesn't also have an STD, using a latex condom, or being vaccinated, but this only protects against some STDs. Now, I'm not going to go through all of these, but I want to mention the sort of general categories of these. And so you've got viral diseases. HIV and AIDS is definitely the most serious one, but you can actually get cancer caused by the human papillomavirus. Now, the 2 diseases caused by HPV, by herpes and by hepatitis B. With HPV, there's definitely a vaccine out. I believe, I'm not sure, but I believe there may be a vaccine for herpes, but I don't think there is for hepatitis. So you basically have the possibility of a vaccination with some of the viral STDs. With the bacterial diseases, there is not going to be a vaccine available. However, these diseases can be cured, completely cleared up with antibiotics if they're not antibiotic resistant. There is a protist STD called trichomoniasis, and for this you have to use antiprotozoan drugs, and that can get pretty nasty. There is also a fungal infection, yeast infection, and for this you would use antifungal drugs. Now, usually a fungal infection will result from some sort of change in the pH of the vagina, because that's usually a good guard against either bacterial or fungal infection. So a lot of events occur between fertilization and birth, and that's what we're going to spend most of the rest of this video discussing. So a zygote is simply a fertilized egg cell, and in the pre-embryonic stage. And so it's not just one single egg, it's not just one single cell. Cell division goes on, and at this point it's still a zygote. Once you've had 3 germ layers differentiating into organ systems, then it becomes called an embryo. Once organ systems continue to develop and become functional, this is where you're going to start calling it a fetus. So in the pre-embryonic stage, you've got a morula, which is a solid ball of 16 or more cells. Now that solid ball begins to hollow out into an outer layer and an inner layer mass, and that's called a blastocyst, and that's what actually implants into the endometrium about 1 week after fertilization. So by 1 week, you've already gone from the 1-cell stage to a blastocyst. A gastrula has 3 germ layers: endoderm, mesoderm, and ectoderm, and we'll talk about those in a little bit more detail in just a bit. Actually, we have talked about those this semester, I believe. That endoderm is responsible for producing internal organs and viscera, that the mesoderm is responsible for producing the heart and skeletal muscle, and that the ectoderm is responsible for producing nervous tissue and skin. So these are the 3 primary germ layers, and when those develop and differentiate into organ systems, this is the embryonic stage. So during fertilization, a number of different events occur. First of all, a sperm cell will meet an egg cell. And by meet an egg cell, I mean it'll come into contact with it. It won't come up to it and say, "How you doing?" So a sperm cell contacts an egg cell. And in this first event here, the sperm will squeeze between the follicle cells that are surrounding that egg cell. In the second stage, the acrosome will burst, and this releases digestive enzymes. And what these will do is to digest through any follicle cells that are still in the way and through the jelly layer that surrounds the egg cell. In the third stage, the sperm nuclear membrane, or I'm sorry, the sperm cell membrane fuses with the egg cell membrane, and this causes the release of the sperm nucleus into the egg cell. And in the fourth part, or in the fourth stage, the sperm nucleus actually fuses with the egg nucleus, and this will form a diploid nucleus. So all the way up until that fusion occurs, fertilization is not complete. Now, a day after fertilization, the zygote begins what's called cleavage, and this is a period of rapid mitosis, rapid division. So cleavage is going to result in a morula, which you see in that first red square, and a blastocyst, which you see in the second red square. The outer layer of the blastocyst is called the trophoblast. These cells become part of the placenta. The inner cell mass actually gives rise to the embryo proper. So about a week after fertilization, the blastocyst will implant. It becomes embedded in the uterine lining. During implantation, the blastocyst will actually digest the uterine lining and obtain nutrients from it. It will also secrete a hormone called human chorionic gonadotropin. And what this does is to preserve the corpus luteum. So you'll recall that if fertilization doesn't occur, that the corpus luteum will begin to degenerate within about 10 to 14 days. And when it does so, the amounts of progesterone and estrogen really reduce. So by maintaining the corpus luteum, the blastocyst also maintains higher levels of estrogen and progesterone, and this prevents the woman from entering another uterine cycle. Now, again, because menstruation occurs only if the corpus luteum degenerates, this secretion of human chorionic gonadotropin helps keep the uterine lining intact. Now, after implantation, the inner cell mass is going to flatten and form the embryonic disc, which you can see at the bottom of this diagram. It's the fourth label up from the bottom of the embryonic disc. And this embryonic disc is going to contain the endoderm, the mesoderm, and the ectoderm, which are all going to develop into different parts of the embryo. So, sorry, I just mentioned that. You form three germ layers: ectoderm, endoderm, and mesoderm. So over the next few weeks after this, the blastula develops into a gastrula and then an embryo, and there are four membranes that will protect, support, and nourish the embryo. And you can see them listed here: the allantois, the yolk sac, the amnion, and the chorion. So the yolk sac, the job of the yolk sac is to manufacture blood cells. At this point, the embryo doesn't have bone marrow with which it can produce blood yet. So the yolk sac will manufacture blood cells very early on. An outpouching of this yolk sac will form the allantois, and this will give rise to the blood vessels of the umbilical cord. So the umbilical artery and the umbilical vein arise from the allantois. The amnion will contain fluid that will cushion the embryo, and it'll also help maintain a constant temperature and pressure. So if you've ever heard of amniotic fluid, this is the fluid that is within the amnion, and if you've ever heard of an amniocentesis, it is a sample of the amniotic fluid that is taken. Now, fetuses shed cells just the same way that we do. We constantly shed cells, and so if you withdraw a sample of amniotic fluid, you are virtually certain to get embryonic cells out of it as well that have been sloughed off from the embryo. And you can look at these cells to see if there are any chromosomal defects, and a lot of the time this is a good idea with high-risk pregnancies. The outermost membrane, the chorion, produces what's called chorionic villi. And these are finger-like extensions that project into the uterine lining, and you see them pointed out on the right-hand diagram, the chorionic villi containing fetal blood vessels, and these actually exchange nutrients with maternal blood. Now, they don't make direct contact, and the fetal blood vessels don't directly attach to the maternal blood vessels, but they're close to one another, and this is going to allow for an exchange of nutrients. So the chorionic villi eventually develop into the placenta, which is the organ that connects the fetal and maternal circulation. And the placenta connects to the embryo through the umbilical cord. Organ formation begins in the 3rd week of development. So the first way that this starts is that a furrow develops, or a depression develops, on the embryonic disc called a primitive streak. Now, this is sort of a line around which other structures organize and develop. This primitive streak gives rise to the notochord, and this will form the framework of the vertebral column. Now, you may remember from way back in January that we talked about our phylum, Chordata, and we said that having a notochord is one of the 4 characteristics of being a chordate, and so the notochord will exist in the embryo, but not later on. So the notochord causes the development of the central nervous system, and the neural groove is your first the step in nervous system development. So if you look at the diagram on the upper right, you'll see where it says neural folds and neural groove. This is your first step in nervous system development. If you look at the lower left, you'll see that the neural groove folds into the neural tube, and this is what actually becomes the brain and the spinal cord. During the 4th week of development, the heart begins beating, and immature lungs and kidneys appear, and so do facial features. So if you look at the bottom right, that is facial features, so you can see an eye there. It's not something that you would consider to be certainly a mature face. In weeks 5 and 6, you see fingers and toes develop. So sex differences will become apparent between weeks 7 and 9 after fertilization. Now, what this shows you is stages of development, okay? So SRY is the sex-determining gene. So if that gene is present, then development will continue as a male. Otherwise, you get female genitals developing. So you can see fused urogenital folds in both. You can see a urethral groove in both. On the left with the male, you'll see a developing penis. On the right, you'll see, in the female, you'll see a developing clitoris. So basically, a penis is a modified clitoris. So you'll also see colored in purple the labia majora on the bottom diagrams, and you'll see both the scrotum and the foreskin. And so the scrotum and the foreskin are basically structures that are adapted from what would have become the labia majora. So if this gene gets activated in week 7, you see the development of male genitalia. If it's not present, or if it's inactive, you'll see female genitalia develop. And so you can have instances where you'll have an individual that either has a defective SRY protein or defective receptors for SRY. And so even though this individual is genetically male, that individual will develop as a female and have a female body plan. Okay, week 9 is the beginning of the fetal stage of development, and it's during this stage that organs develop, that bones replace cartilage, and nerves and muscles start to coordinate their actions. So you see the fetus at 9 weeks, 13 weeks, and at 20 weeks. Near the end of the pregnancy, the digestive and respiratory systems mature, and neural connections start to form in the brain, and after about 38 weeks is when a baby is ready to be born. Birth is a 3-step process. The first one is labor, and in this begins with the amniotic sac breaking, and hormones will start to prompt uterine muscle contractions. The baby's head will press against and open the cervix. Now, it's when the cervix gets stretched out that the posterior pituitary is directed to produce oxytocin. So what that will do is to increase the strength of the muscle contractions. The baby's head presses against the cervix harder. The cervix gets stretched out harder, sends more signals to the central nervous system. The hypothalamus says, "Oh, you're being stretched out. Have some more oxytocin." And so that continues to amplify until you get to delivery, which is when the baby is actually descending through the vagina. In the third step of birth, the uterus will expel the placenta. And so there's your three steps: labor, delivery, and the expelling of the placenta. Now, as the baby emerges, I mentioned this before, the cervix will get stretched further and further out. This produces more and more oxytocin, and this intensifies the contractions to a greater and greater degree. And this is an example of positive feedback, where production of the hormone results in consequences that result in more production of the hormone, which results in consequences that produce even more. So this is kind of a snowball effect. Okay, so from here we're going to talk about some birth defects. There are a lot of causes of this. So about 3% of newborns have a birth defect that results in some sort of disability of the child. So in the previous picture, it looks to me like that was someone who had been exposed to something called thalidomide. And I'll talk about that more in just a minute. So teratogens are substances that cause birth defects. Chemicals and metals can do this. And so thalidomide, a chemical that I was talking about, this was something that was administered to pregnant women as a way of treating morning sickness. And so apparently it worked very well, but the problem is what did not show up in animal trials is that this drug specifically disrupted the formation of limbs in humans. And so you saw people who were born with limbs that didn't have as many fingers. Sometimes just a couple of fingers. The limbs would be really shortened, and sometimes would be almost flipper-like. So that's where, so that would come under the heading of prescription drugs. And if you look at the very bottom graph there, you can see that thalidomide is only acts during a short time during the pregnancy, but if it's being taken during that time, it will cause those birth defects. So chemicals and metals, alcohol, cigarettes, illegal drugs, prescription drugs, vitamins, viruses, all of these things can cause birth defects. So last, we're going to wrap this up by talking about something that a lot of insects do that I'm really glad that we don't do. So this is the ultimate sacrifice. So the two spiders are the same species, and the male is a whole lot smaller. And it has about a 2 in 3 chance of being eaten by the much larger female. So it has a 2 in 3 chance of being eaten by the much larger female. Now you might think that the male would try to avoid being cannibalized, but during mating, he actually somersaults into the female's mouth, offering his body as food. And I think that the wording here is kind of funny, because I get this picture of the male somersaulting into the female, and on the side you've got the East German judge gives him 6. But why would natural selection favor this kind of really weird behavior? Well, researchers set up experiments to find out. And each copulation between spiders lasted between 6 to 31 minutes. But males that were ultimately cannibalized spent about 25 minutes in the act, and males that survived spent about 11 minutes in the act. So a longer copulation resulted in more fertilized eggs. So if a female ate her first male, she rejected another male 67% of the time. But if the male survived and got away, the female didn't reject the second male very much, only 4% of the time. So the males who get eaten have, first of all, a higher chance of getting the female pregnant in the first place. But they all have a much, much greater chance of the female refusing a subsequent mate. And so sexual reproduction is really all about kind of getting genes into the next generation. And so natural selection is going to select 4 behaviors, no matter how bizarre, that give the parents a better chance at getting their genes into the next generation. And so that's the reason why the males tend to do like a double gainer into the female and say, oh, here you go, first meal. So sexual cannibalism boosts the male spider fitness in a couple of different ways. Longer copulation time plus fewer mating opportunities for males that come along afterwards. And again, because these cannibalized males were much more likely to pass on their genes, that's what natural selection favored. So what this is a really big reminder of to you, and this is a great thing to remember heading into our test 5 material, is that natural selection does not favor survival. It selects for strategies that increase reproductive success. So with male spiders being likely only to have one opportunity to mate, any behavior that makes this one opportunity a success gets strongly selected for.